Hello, 428s and Brian. I hope you all had a relaxing, lovely reading break. My name is Kyle Richards, and today I'll be sharing my presentation on Aequorea victoria, or the crystal jellyfish, and how this simple marine organism sparked one of the greatest revolutions in modern biology through its brilliant bioluminescence system. I'll start with a brief introduction and historical background of Aequorea, then I'll explain how its bioluminescent mechanism works at a molecular level. We'll look at how the proteins responsible for bioluminescence have been adapted into powerful tools in biotechnology, and finally, I'll wrap up with where this field is heading in the future. Bioluminescence is the natural production of light through enzyme-driven chemical reactions. It's found in many environments, but is especially widespread in the ocean, where light plays key roles in communication, mating, and defense. Among these marine species, Aequorea stands out, not only for its beauty, but because its light-producing system reveals two proteins that have incredible influence on molecular biology. For Aequorea itself, bioluminescence is thought to function in predator avoidance and communication, using brief blue flashes to startle threats or signal nearby organisms in the coastal waters it inhabits. In the 1960s, Osamu Shimomura isolated the proteins Aequorin and green fluorescent protein, or GFP, from thousands of jellyfish collected at Friday's Harbor in Washington. Shimomura's work was mostly curiosity-driven, but it laid the groundwork for many transformative discoveries that followed. Martin Shelfie later expressed GFP in other organisms, proving that the protein could fluoresce in systems outside of jellyfish. Roger Chien then engineered new variants of proteins that could fluoresce other colors and with improved brightness, leading to a shared Nobel Prize in Chemistry in 2008 for the three researchers. In the image at the bottom, we can see Aequorea's photoorgans on the left, a whole jellyfish in the dark in the middle, and finally another whole jellyfish under visible light on the right. Aequorea's light comes from two coordinated proteins, Aequorin and GFP. Aequorin is made of apoaquorin, the inactive form of the protein, cilantrazine, and oxygen. When calcium binds to Aequorin, it triggers the oxidation of cilantrazine. This oxidation of cilantrazine produces cilantaramide and CO2. Excitation of cilantaramide produces blue light at about 470 nanometers. GFP then absorbs this energy and re-emits it as green light at 508 nanometers through a process called bioluminescence resonance energy transfer, or BRET. This is an example of non-radiative transfer, which means the energy is passed electrically, not through emitted light, which makes it faster and far more efficient. I will elaborate on this concept in the following slide, but this is the general mechanism for how Aequorea converts a simple chemical signal of calcium into its signature green glow. GFP is a beta-barrel protein with a self-forming chromophore made from three amino acids: serine, tyrosine, and glycine. A chromophore is the portion of a molecule that is responsible for color creation. This beta barrel protects the chromophore, ensuring the fluorescence stays bright and stable under varying conditions. In order for energy to pass from the excited cilantaramide to GFP via BRET, two requirements must be satisfied. Firstly, there needs to be light spectrum overlap between Aequorin and GFP. Every light-emitting molecule has an emission spectrum, which is the range of wavelengths or colors it gives off. And every light-absorbing molecule has an absorption spectrum, which is the range of wavelengths it can absorb. For energy to move efficiently from the donor, which is Aequorin, to the acceptor, which is GFP, the light Aequorin emits must fall within the range of light that GFP can absorb. Aequorin emits blue light, peaking around 470 nanometers, and GFP absorbs maximally around 475 to 490 nanometers. Because the spectral overlap is highly aligned, GFP can absorb the energy released by Aequorin without the energy first leaving as blue light. And the second requirement, in addition to spectral overlap, is the two proteins must be close together, typically within 1 to 10 nanometers. The closer together, the more optimal the energy transfer. For this interaction to be successful, the excited donor's oscillating electric field must directly excite the acceptor's electrons. If they're too far apart, that coupling collapses and the energy dissipates as light or heat instead of being transferred. Now that we have discussed the molecular underpinnings of the mechanism, I'm going to introduce some applications derived from the system. GFP is one of the most powerful tools in modern biology because it can act as a genetically encoded fluorescent tag. What that means is that researchers can attach the GFP gene directly to any gene of interest. When that gene is expressed, GFP is expressed alongside it. And since GFP fluoresces naturally, we can literally watch biological processes happen in real time. For example, in a living embryo, GFP can be used to trace how specific tissues or organs form and change over time. In neurons, it can reveal how proteins move along axons or how synapses reorganize from learning. In cancer research, scientists track tumor growth or metastasis by linking GFP to oncogenes. Oncogenes are a mutated or overactive gene that drives uncontrolled cell growth and can lead to cancer. One of the GFP's biggest advantages is that it doesn't need any chemicals or cofactors. It folds and fluoresces all on its own. That's why it works in everything from E. coli bacteria to complex vertebrate systems, including humans. Its discovery completely transformed cell and developmental biology, fostering dynamic and visual experiments that show how life unfolds on the molecular level. Aequorea, meanwhile, serves as a natural calcium sensor. It works beautifully because it directly converts a chemical signal, i.e., calcium, into visible light. Inside cells, calcium levels rise and fall constantly to control processes like muscle contraction, neurotransmitter release, and metabolism. Only when Aequorea binds calcium does it emit light. That means its luminescence precisely mirrors the calcium activity happening inside the cell. What makes Aequorea so valuable is that it doesn't require external illumination like fluorescent dyes or microscopes. This eliminates background noise, giving researchers a clean, reliable readout of calcium dynamics in real time. It's been used to study how heart cells contract, how neurons fire in response to stimuli, and how metabolic pathways fluctuate under stress. Beyond visualization, GFP and Aequorea have become cornerstones of modern analytical biology and drug discovery. To reiterate, Aequorea's bioluminescence has a key advantage, which is that it produces light only in response to calcium binding, which has almost no background noise. This makes it ideal for large-scale, high-throughput screening, where thousands of drug candidates can be tested simultaneously for effects on calcium signaling. It's been especially valuable for studying G-protein coupled receptors, or GPCRs, and ion channels. These are two of the most important classes of drug targets in pharmacology. Just as a refresher, GPCRs are the largest class of cell membrane receptors that are responsible for detecting external signals like hormones and neurotransmitters. GFP complements this by providing real-time fluorescent readouts of cellular behavior. By attaching GFP to specific genes, proteins, or receptors, researchers can monitor processes like gene activation, protein movement, and receptor internalization with greater clarity. Together, these systems allow for automated, high-precision measurements that use light as a quantitative readout of molecular activity. One of the biggest challenges faced today is that blue and green light, like that from Aequorea and GFP, doesn't penetrate tissues very deeply, limiting how far we can see into complex organisms. To overcome this, researchers are developing red-shifted or near-infrared variants of fluorescent proteins that can illuminate deep tissues. These new proteins open the door for imaging processes in the brain, heart, and other internal organs without invasive procedures. Another exciting development is the rise of new engineered light-producing enzymes, like NanoLook, which are smaller, brighter, and more stable enzymes that outperform natural systems. These are helping scientists visualize molecular activity at single-cell resolution and track long-term changes without signals fading. The future also points towards integrated biosensors that activate only under specific physiological conditions. For example, lighting up in responses to changes in pH, or changes in oxygen levels, or specific metabolites. These smart systems will improve precision and have minimal background interference. In summary, the crystal jellyfish, or Aequorea victoria, provided the molecular foundation for a revolution in biological imaging. Its two key proteins, Aequorea and GFP, represent distinct yet complementary mechanisms of light emission. Aequorea acts as a calcium-sensitive photoprotein, converting transient changes in intracellular calcium into measurable blue luminescence. GFP functions as a genetically encoded fluorescent protein, or reporter, allowing researchers to visualize gene expression, protein localization, and cellular dynamics in real time. Together, these molecules continue to advance microscopy and biosensing, enabling direct observation of physiological processes that were once invisible. As engineered variants become brighter and more photostable, optical imaging is extending into deeper tissues and more complex systems. The significance of Aequorea now extends beyond marine biology. Its bioluminescent system has become an indispensable tool for understanding the mechanisms that govern life at a cellular and molecular level. That is all. Thank you so much for listening. I hope you guys have a great weekend.